All right, let's talk about medical news. We see it every day, right? Headlines about some miracle drug, some huge breakthrough. But how much of it should we actually believe? Well, today, we're going to give you the tools you need to become a much sharper, more critical reader of all that news. We're going to do that by really digging into the gold standard of medical evidence, the clinical trial. So, to get us started, check out this great story from an oncologist, Dr. Vinay Prasad. He had a pre-med student shadowing him who also happened to be a talented artist. A little while later, she came back and gave him this charcoal portrait she'd drawn of him. Super nice gesture, right? Very flattering. But it wasn't exactly accurate. As Dr. Prasad put it, the student took a few artistic liberties. So what were these artistic liberties? Well, the portrait showed him in a white coat, which he doesn't own. It gave him a stethoscope, but as a cancer doctor, his main tool is actually a PET scanner. It showed him scribbling notes, but he prefers to just listen and remember. You see, the portrait looks like what we think a doctor should look like, but it's an idealized version, not the real thing. And that brings us to the whole point of this explainer. A clinical trial is just like that portrait. It's a representation of medical truth, and it's designed to be as accurate as it possibly can be. But, and this is a big but, it isn't the truth itself. To really get what a trial is telling us, we have to learn how to spot those artistic liberties, all the little choices in how a study is designed that can bend and shape the final picture. Okay. So before we start looking for all the ways the picture can get distorted, let's first figure out what the ideal portrait is supposed to look like. What is this gold standard of medical evidence anyway? It's called the randomized controlled trial, or an RCT for short. The idea is pretty simple. You take a group of patients, and then you randomly assign them to one of two groups. One group gets the new treatment you're testing, and the other group gets a control. That could be a sugar pill or maybe the best treatment that's already out there. The whole goal of randomizing is to make those two groups as identical as possible. That way, at the end of the day, any difference you see in their health, you can be pretty darn sure it was because of the new drug. That randomization part is absolutely key. Now, here's something that trips a lot of people up. The word random can mean two totally different things. On one hand, you've got random sampling. That's what political pollsters do. They call a few hundred people who are supposed to represent the entire country. The goal there is generalizability. But clinical trials, they almost never do that. Instead, they do something called random allocation. They basically take whoever they can get to sign up for the trial, what's called a convenient sample, and then they randomly split that group. This doesn't mean the results apply to everybody, but it does mean you get a fair fight between the two groups in that specific trial. And that difference is critically important. And that leads us perfectly into the very first question in our Skeptics Toolkit. Who exactly are the people in this convenient sample? So since the people in the trial aren't a perfect slice of the real world, you have to ask yourself, do they even look like real-world patients? Every trial publishes a Table 1 that describes who they are, but do they match up? For instance, the average age someone gets diagnosed with lung cancer is 71. But you look at the trials, and the patients are often way younger. They have all these strict rules that kick out patients who are maybe too old, too sick, or have other medical problems. So you have to wonder, if the people in the trial don't look like the people in the real world, how well are the results really going to apply? All right, on to question number 2. A trial is a comparison, right? It's a matchup between the new kid on the block and something else. So we have to ask, was it a fair fight? This is one of the biggest and maybe one of the most clever artistic liberties you'll find. It's surprisingly easy to make a new drug look like a superhero just by putting it in the ring against a really weak opponent. The amazing benefit you see in the headline, it's only as real as the thing they compared it against. So, how do you spot a weak opponent? What we call a weak control arm. Well, sometimes they'll compare the new drug to a placebo, a sugar pill, even when there's already a good treatment that exists. That's a huge red flag. Or maybe they give the control group the right drug, but at a dose that's too low. And here's a really subtle one. They give bad post-protocol care. That means they don't give the best possible treatments to the control group after their disease gets worse during the trial. Let me give you a wild example from a real trial for a myeloma drug called daratumumab. So you have the control group, and when their cancer gets worse, they're supposed to get the best care available. The obvious next step for them would have been, well, daratumumab. But get this. In the trial, more than half of them never got it. The company running the trial, the one that makes the drug, they just didn't give it to the control group. Of course, the new drug combo looks better, but that doesn't answer the real question, which is it better than just using the drugs one after the other? Okay, on to our third and final question. We know who was in the trial, we know who they were up against. Now we have to ask, what was the scoreboard? How did the researchers actually decide who won? And this is where things can get really tricky. There's what matters to patients, and then there's what's easy for researchers to measure. The ultimate goal, the thing we all care about, is overall survival, or OS. Did the drug help people live longer? It's simple, it's objective, it's what matters. But it takes a really long time to measure. So a lot of trials use a shortcut, a surrogate endpoint called progression-free survival, or PFS. This just measures how long it takes for a tumor to get bigger on a scan. It's faster, but as we're about to see, a better PFS doesn't always mean a better or longer life. Let's look at the Bolero trial. This was for a breast cancer drug. The results for progression-free survival were incredible. I mean, just look at that graph. It showed a huge benefit. Based on this data, the drug got approved. It looked like a total game changer.
But then, a while later, the overall survival data finally came out, and there was nothing. No difference. The drug didn't help people live any longer. So wait a minute, how is that even possible? How can a drug be so good at stopping a tumor from growing on a scan, but have absolutely no effect on how long a person lives? It just doesn't seem to make sense.
Dr. Prasad has the absolute perfect analogy for this. He says, imagine he sells you a $12,000 energy drink before you run a marathon. You drink it and, wow, you run the first 7 miles faster than you ever have in your life. But then you completely hit a wall. You crash and burn, and you have to drag yourself through the rest of the race. You end up crossing the finish line at the exact same time you would have anyway. Would you be happy? No. You'd say, why in the world did I just spend 12 grand on your drink to get the same time? That's PFS versus OS. A great start, but the same finish. So what's really going on here with the stats? Well, one of the biggest reasons is a hidden bias called informative censoring. It sounds complicated, but the idea is simple. If a new drug is, to use Dr. Prasad's words, toxic as all hell, what happens? The sickest patients, the ones who are most likely to have a bad outcome anyway, they're the first to drop out of the trial. They just can't handle the side effects. So the only people left are the toughest, healthiest patients who can stick it out. This makes the drug look way better than it actually is, because you've essentially filtered out all the people it was hurting the most. So we've seen it. The portrait of a clinical trial can be distorted in all sorts of ways, by choosing who gets painted into the picture, by who they're compared against, and by what you decide to call a masterpiece. So let's put it all together. Here it is, your toolkit. It's just 3 simple questions. Number 1, who was in the trial? Do they actually look like real patients? Number 2, how was it tested? Was the comparison a fair fight against the best available option? And number 3, what was measured? Was it something that actually matters to a patient, like living longer, or was it just a shadow on a scan?
Ultimately, asking those 3 questions gets you to this one final, powerful question you should ask every single time you read about a new study. The result might be statistically significant, meaning the math says it's probably not a fluke. But our job as smart, critical readers is to ask, is it clinically significant? Does this number, this result, actually make a meaningful difference in a person's life? Thanks for watching The Explainer.